All right, there we go. We're back. All right, it's good to see everyone back this morning. We haven't actually met in person since last Friday. Been a cold, dark 5 days for everyone, I'm sure. Actually, it's been really nice out. I have not checked in on the exam scores yet, so I don't have those results to present for the class, but I will get those to you. We should be able to look at those on Friday. Just as a reminder, lab materials, your skull drawings, just hang on to those. I'll look at them in person in lab. A few of you emailed them to me, which I appreciated, but what I want to do is just sit down with everyone individually and make sure you've got it, make sure it makes sense, and we'll check it off that way. I want to remind you that you have quizzes in visible body on the skull that were due, well, they were due yesterday, and then you have quizzes in Blackboard on the special senses, which are due before next week's lab. If you didn't get to those skull quizzes in visible body and need to do it still, just shoot me an email and I'll open it up again. We can just be flexible. I wanted to have you do it before you got the special senses lab due, so you didn't have like a whole bunch of lab stuff piling on. But if for some reason you didn't get to it, just let me know. All right, so today we're going to continue with the brain. This is probably, well, we're not going to get through the brain today. We'll probably wrap up the brain on Friday. And so our agenda looks like this. We're going to review Friday's ticket out. Make sure you've got some understanding of those concepts. We're going to finish up talking about the thalamus. I've got a review activity involving drawing, which you're super excited for, I know. We're going to start in with the cerebrum and just see how far we get today. And we'll finish up whatever we don't get to today on Friday. I've got a wrap up and a ticket out for you. So on Friday, when you're leaving class, I asked you this question: which of the following are a part of the diencephalon? Select all that are true, and boom. The class rocked this one. Remember, the diencephalon is composed of the thalamus, hypothalamus, and epithalamus. Now, I know we didn't talk about the hypothalamus and epithalamus in any detail yet. We just started on the hypothalamus. Make sure you know that those three together form the diencephalon. Class, looks like you got that one all good, so nice work there. The next question is, where do most of the cranial nerves connect to the brain? Remember, we have 12 cranial nerves. We haven't talked about in too much detail, but when we were talking about the brain stem, we did emphasize that 10 of the 12 connect to the brain via the brain stem. So about half of you got that. There's 2 that connect directly from the cerebrum, and none of them from the diencephalon or the cerebellum. So make sure you know that those cranial nerves are connecting primarily through the brain stem. Brain stem, remember, is composed of your midbrain, pons, medulla. Sound good so far? All right, one more question. Oops, actually, that is not supposed to be like that, but that's okay. I gave you this thought experiment. Imagine you're a tiny organism and someone asks you for directions to is it the superior colliculi? Let me just fix this slide for 1 second for you. Well, it got cut off, actually. I apologize. I think it was the superior colliculi there. And this one, the correct answer was actually orange. So less than half the class got this. And I just want to walk you through this. Remember, if you're going to those superior colliculi, you can go, if you find the diencephalon, which is where the thalamus primarily, right? And then you move inferiorly to the midbrain. And then once you're in the midbrain, the superior colliculi are in the dorsal aspect of the midbrain. So the directions would be to find diencephalon here, move inferiorly or caudally to the midbrain, which is here, and those colliculi are on the dorsal aspect of the midbrain, so between the midbrain and the cerebellum. Remember, you have 4 of those colliculi. You have 2 superior and 2 inferior. So I just wanted to review that, just kind of jogging your memory a little bit about where those colliculi are. Questions you have on any of those? Okay, well, let's jump back and talk about the brain. This is the schematic that you have memorized at this point. We've talked about the brain stem already. We talked about the cerebellum already, and now we've moved on to the diencephalon. Note that the 3 primary structures of the diencephalon, the thalamus, hypothalamus, epithalamus, all 1 have the word thalamus in them, and 2 are adult structures that develop from the same embryonic or developmental structure. All 3 of those structures emerge from the diencephalon, And so we still call this structure both embryonically and adult brain the diencephalon. And you see it, it's this purple bit kind of right in the center of the brain. Remember that the cerebellum is covering this. We're actually seeing through the left hemisphere of the, excuse me, I said cerebellum, the left hemisphere of the cerebrum here. So this diencephalon is essentially like right smack dab in the middle of the brain. And we talked about this a little bit on Friday. I just want to review a couple slides and then we'll get to some new information about it. Specifically, we have the thalamus. Oops, the majority of the diencephalon is the thalamus, this big thing here. Kind of in the back, inferiorly, we have this little guy hanging off, the epithalamus, and then in the front, kind of anteriorly and inferiorly, we have the hypothalamus. And remember that off the hypothalamus, we have this very, very important endocrine gland that's very intimately connected with the hypothalamus, and this is what pituitary. Good. And the pituitary sits in a little saddle in a bone called the the sola-turcica. And what bone is that in? Sphenoid. Yep. Good. Cool. So this is, if we look at the diencephalon in a little more detail without other structures around it, pituitary is here, and we have this infundibulum that connects the hypothalamus and the pituitary. And infundibulum contains blood vessels that carry hormones, and also contains axons of neurons that transmit transmit signals directly between the pituitary and the hypothalamus. So the pituitary and the hypothalamus are kind of this real crux of where the nervous system and the endocrine systems are integrated. Got you. Focus, right. There's a lot of different reasons for that, and I don't know the details. That's a Dr. Vetney question. Like what parts of the brain allow you to be hyper-focused versus easily distracted? And there's environmental factors like in a given moment that can affect that. Like, you know, we all know we have days when it's easier for us to concentrate and easier for us to get distracted. So it changes within an individual, but then also different people have patterns for that. And there's a lot of complex reasons for that are beyond the scope of what I can tell you, unfortunately. I'm going to just make a list of like Dr. Vadney questions. So let's think about the anatomy of the diencephalon. Remember that the thalamus is paired. And so if we look at it from an inferior view, it looks like this. Notice we've got some of the cranial nerves coming off of the midbrain here. And we actually have two of them that are run from the cerebrum right here, the optic nerve. So that is your cranial nerve 1. And you can kind of see where they nestle just inferior to the thalamus. And so if we take a look at this, I think this is where we left off on Friday, talking about all of these different nuclei, these centers of gray matter in the hypothalamus. Again, you do not need to know all of these and what they do. Rather, I want you to know that there are all of these nuclei in the hypothalamus. And just as a reminder, those nuclei, it's gray matter that's composed of the cell bodies of axons that extend out of the gray matter, but also small unmyelinated interneurons that are connecting the cell bodies of those other neurons connected to each other. And so we have these different centers. They look a little bit like water balloons here, but they're not really look like that. So it's color-coded so you can see them. And they're involved with a whole bunch of autonomic functions of the body, everything from control of like your urges to drink and eat, body temperature regulation, sex drive, all of those things are regulated by nuclei in the hypothalamus in conjunction with other parts of the brain. Now, if we look at this on an actual brain, I actually like this diagram a lot because you can see what it looks like. You can see the brain, the medulla here, the pons, the midbrain is kind of up here, though you can't see it quite well. You can see where everything is located. We'll see this in more detail in the lab. Next week, Tuesday, we have the brain lab. So just want to show you what that actually looks like on an actual brain. And we'll get into that in detail in the lab. Let's see. Did I do this slide on Friday? I don't remember if I did this one. Okay, So I just kind of got ahead of myself a little bit. What I'm going to do is just go ahead and put these up and I'll walk through them. As I mentioned, the nuclei in the hypothalamus control a number of autonomic nervous system functions. I gave you some examples. What you want to note here is it's things we're not thinking about consciously. So heart rate, blood pressure, salivary glands, which is kind of interesting. Also ties to emotional response, especially kind of extreme emotions, things like rage or fear or pleasure. Body temperature regulation, I mentioned, so centers that regulate whether our two primary mechanisms of regulating our body temperature are sweating to cool down or shivering to warm up. Hunger and thirst. Oh, I didn't mention this one, sleep-wake cycles, although that is also controlled by your pineal gland and epithalamus, we'll talk about in a little bit. And then the hypothalamus is kind of a master control center of your endocrine system. Feeding and sex drive I talked about. And then there is some memory formation involved with the hypothalamus as well, although it's not really its primary function. And that is primarily in the mammillary bodies. So let me show you where those are. I'll come back to this slide if you're still copying. I just want to show you those mammillary bodies on the previous slide. Mammillary body is these kind of bulbous structures right here. And if I flip ahead one slide, you can see them, like, for example, mammillary bodies, this guy right here, just anterior. Let me let you get that down. All right, the final part of the diencephalon is the epithalamus. And it is called epi because, not because it's on top of the thalamus, but because it's on top of this third ventricle, which is where the cerebrospinal fluid is. So it kind of sits kind of right here. And what I really want to focus on is the pineal gland that is part of the epithalamus. And the pineal gland is actually regulated by the hypothalamus. So there's a lot of crosstalk there. But the pineal gland is primarily involved with secreting the hormone melatonin, and that helps us regulate sleep and our circadian rhythm, and it suppresses your gonadal function when it's dark. So interestingly, and kind of contrary to our modern societal practices, our intrinsic sex drive is actually regulated in contraposition to our sleep drive. It's such that the sex drive is being suppressed at night in the dark when melatonin is released, and then vice versa. So there's some information for you. And that's all we're going to say about the epithalamus. It's a little guy in the back. It's located just above the third ventricle. It's got a pineal gland. It's important for sleep. Melatonin. That's the other word. Notice I did say that there are nuclei in the hypothalamus involved with regulating sleep, and those guys are cross-talking with the hypothalamus and the pineal gland. So there's multiple layers of regulation there. We won't get into those details in this class. We'll save that for your human phys class, or behavioral endocrinology actually would be the class for you to really get into the details of how these systems work. All right, that is the diencephalon. I want to transition now and talk about the cerebrum, but before we do, guess what? Yes, Taryn, nice job. How did you know? Try to be so like, yeah, I know, try to be surprising. All right, this is going to be a bigger one, but as best you can from memory, of course, we'll go back and confirm your notes. Draw me a brainstem and then label the midbrain, pons, and medulla add the cerebellum, add a diencephalon, label the thalamus, hypothalamus, and epithalamus, throw in the pineal gland, and just for fun, add the pituitary. So I'm not looking for artistic perfection here. Kind of do like a schematic, use some basic shapes to draw these out. Don't worry about getting the exact shape. What I'm really interested in seeing in these diagrams is the relationship of the positions of these parts of the body relative, or parts of the brain relative to each other, as well as kind of the relative size. So if you draw me epithalamus and it's the same size as your cerebellum, that's kind of showing me you're not getting those kind of scale relationships. Although I don't need to know exactly the precise relationship, but just generally what's big and what's small. So I'll give you a few minutes to do that. I don't have a timer on this slide, but I'll just kind of monitor when I see people done drawing, and we will come back and show you what that looks like and talk about it. I think most people will wrap up their diagrams. Let's go ahead and go back and let me give it. I see people drawing. Let's give another 30 seconds. Finish up your drawings. All right, so your diagram should look something like this. So this is a brain from the lateral view, cut mid-sagittally. You've got the big old cerebrum. You've got your thalamus here, hypothalamus, pituitary, epithalamus back here, cerebellum back here, and your midbrain, including your your brain stem, including midbrain, medulla here. And of course, if you label this part down here, what would this be? Spinal cord. Good. So remember that the medulla is continuous with the spinal cord there. Yeah, you can see some other features in here. The corpus callosum, actually, which is something we'll talk about in a minute. This is the center of white matter connecting the hemispheres of your brain. And of course, your ventricles as well, where that cerebral spinal fluid lives. Any questions on that before we get to the last and largest and most complex structure of the brain? All right, let's talk about your cerebrum. So this is by far the largest part of the brain. As we work through this, I've got a study guide for you here as well, similar to what I had for the cerebellum. So I'll let you give you a second to pull that up. It's got the standard diagrams and that kind of thing. The cerebrum is about more than 80% of your brain, and it has this really kind of unique and characteristic texture and shape. And in fact, most of us, when someone just says the word brain in the vernacular sense, we probably are just thinking about the cerebrum. The cerebrum is characterized by these fissures between these different folds, and the fissures separate the two hemispheres from each other, as well as the different parts of this, as well as the cerebrum from the cerebellum here. We've got a fissure that goes right down the middle, not surprisingly called the longitudinal fissure, and that separates the left and right hemispheres. And we have this fissure called the transverse fissure that separates the cerebrum from the cerebellum laterally. Now, the brain is characterized by these kind of like winding, folding shapes that are actually super important for how the brain functions. And we describe these in a couple different ways. As I said, a fissure is a really, really deep groove. We find that between the hemispheres, as I said. But these folds and the grooves between them are called, in the singular, a single fold is called a gyrus, and a single groove is called a sulcus. In plural, they're called gyri and sulci. And that's basically this up and down pattern of the surface of the cerebrum. Now, what is important about these folds is that they serve to increase the surface area. So if you have something that's smooth and you have its surface, it's going to, you're just going to have that outer layer be the surface. Now, our cerebral cortex is this gray matter on the outside, and it follows these sulci and these gyri like this, so that having all these folds allows the brain to be the same size, but greatly increase the actual surface area of the cerebrum. And since our cerebral cortices are in the surface area, it thereby increases the size and the mass of those cerebral cortices. Mm-hmm. Yep. This one? Yep. Mm-hmm. So we've got fissures, and we've got sulci and gyri. Yeah. Okay. Good. Thanks, Kyle. Mm-hmm. Mm-hmm. Yeah, and so a fissure is essentially just a deep sulcus, and that's perfect segue to the next slide. So some of those sulci are deeper, and these deeper sulci divide the cerebrum into these different folds. Now, I'm going to say, do I want to call those fissures? No, I'm going to reserve the word fissure for the longitudinal fissure between the hemispheres and the transverse fissure between the cerebrum and the cerebellum. Note that a fissure is just a really, really deep sulcus, right? There's our other deeper sulci that aren't quite as deep as a fissure, and those are going to divide the cerebrum into pieces. Am I explaining that? Is that me? And we have the lobes very conveniently are named the same as the skull bones that overlay them. So these terms should be familiar. We have the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. Note that they are a left and a right of each of these as well. So we're looking at this brain from a lateral view, but of course, there's a left and a right frontal lobe, left and a right parietal lobe, et cetera, et cetera. Now, when we look at this is what it looks like in an actual brain. If we go to a diagram, we can see that a little more clearly because we can color code it. There's a couple things I want you to take away from this figure. The first is that this big deep sulcus right here between the frontal and parietal lobes is called the central sulcus. And it is important in demarcating those two lobes of the brain. Just anterior to that, we the central sulcus, we have the precentral gyrus, which is aptly named. It's just before the central sulcus. And then we have the postcentral gyrus, and that's part of the parietal lobe you see there. We then have a deep sulcus between the parietal and occipital lobes called the parieto-occipital sulcus, not surprising again. And let me see, is there any other, and I want to show you one more thing on this figure. Actually, I'll do that on the next figure. So we have frontal, parietal, occipital, temporal lobe, that's 4. We have one more lobe of the brain that's not named it's not named in conjunction with the skull bone because it's deep. And there is something called the insula. The insula is deep in the lateral sulcus. So you can see here that there's this tool that's opening up this temporal lobe so that you can see that insula lobe inside of there. So each hemisphere has 5 lobes, 4 of which you can see from the outside, one of which, the insula lobe, you can only see when you are doing a dissection. Now, on an actual brain, these deeper sulci aren't always super obvious. And you'll see this when we get to lab. But it is important to note that some of the structures, that you know where these structures are generally. So, if I point to this area, you might not know exactly where that central sulcus is between the frontal lobe and the parietal lobe. But you know that the frontal lobe is up here, parietal lobe is back here, occipital lobe is back. The fissures, on the other hand, are super obvious. Like that fissure that separates the left and right hemisphere. You will not mistake that in our brain. That's quite easy to see. With that in mind, let's see. I'm going to talk about a little bit generally of the functions of each of those lobes. I'm not going to get into this in too much detail at all, but just give you a general idea what those different lobes are doing cognitively. But let me pause here before I get into that and see what questions you have. All right, let's talk first about the frontal lobe. This is I guess you call it the lobe where we think about where we're the most self-conscious and where like our sense of self resides. So this is where you're making plans, you're initiating actions, you're especially in relation to vision and speech, so important communication center. You're also kind of doing this higher order thinking. So this is the area that's lighting up when you're studying for an exam, or you're thinking seriously about what your internship you're going to do this summer, or where you're going to go on your next date, or whatever. That stuff is happening up in the frontal lobe. And this is the area of the lobe, just as a little aside, when they do an operation called a frontal lobotomy. This is the area of the lobe. This is the area of the brain that they ablate or kind of destroy, and that's why that operation often has such dramatic effects on people's personality. I think in some very limited contexts they do. I'll have to go on an aside one day. I don't want to do it today. There was a guy in the early 20th century who went on tour around the US with an ice pick, literally, and like went to places where they treated quote unquote mentally deficient people and things of that sort. And they would literally go in with a modified ice pick and stick it up their nose and wiggle it a bit and fuck up the frontal lobe. Sorry. It was terrible. Like, it was super terrible. And then they'd be like, all right. And like lots of people ended up dying, and lots of people end up with like serious negative repercussions. But this guy was like super popular and would go like city to city, and people would bring their kids to them. John F. Kennedy's younger sister actually had that. I don't know if it's the same guy, but had a frontal lobotomy when she was really young against their mother's knowledge. I think it was, if not against her knowledge, against her will. Like they didn't even know, and they like took this little kid and they like it's really wild. If you had some of the history of like medical history in general is pretty wild, but the mental health and kind of our understanding of like neuroanatomy and that kind of thing is even more crazy. So yeah, but I think there are some very specific limited contexts now where an operation, obviously they don't use an ice pick, where some more targeted kinds of these surgeries can happen, but it's not. Sorry, I didn't mean to go off on that tangent. I did anyway. Parietal lobe, that is especially important for spatial awareness. So it's integrating information about where you are in space. So information from your joints, information from your visual centers, et cetera, et cetera. And also contains a center for understanding speech. And what's kind of cool is the centers for producing speech and the centers for understanding speech are in two different places. So if you have an injury or something happens to one of those centers, it can negatively impact one thing or the other. So in one case, you might be able to talk perfectly fine, but you have a hard time recognizing what other people are saying. And if you injure the other area, vice versa, you can understand things just fine, but you can't produce speech. Your occipital lobe in the back of the brain, this is important to note, is the center of your visual cortex. So processing all of your visual input from your eyes, that information actually happens in the back of the head. And that's why an injury to the back of the head can actually impair your ability to process visual information. What's interesting in that case is your eyes work just fine. You're still receiving light in your retinas and sending those signals back to your brain. Your brain just doesn't know what to do with that information and doesn't process it in a way that is kind of meaningful or impairs that process. The temporal lobe is the center of auditory, which is not surprising because it's near the ears, and the olfactory cortex, so that is smell, as well as recognition of objects, words, faces, and language. So this is kind of an interesting idea that you can receive information about things, but if there's an injury to this area, you might not know what those things are. So for example, it might be more difficult for you if someone hands you an orange to integrate the smell and the texture and the sight of the object and to put all that information together and know that it's an orange. Processes that we generally not even thinking about. But that's happening in the temporal lobe. Go ahead.
Is that why taste and smell are like so closely connected in the process?
It is part of the reason, yes, and also the way that the signals are integrated. Actually, our sense of taste is pretty weak in the sense that we don't have the ability to discern a whole bunch. But anytime you're eating things, you're also simultaneously smelling it. And so a lot of what we think of as taste is actually due to the smell. And that's where you get the idea of flavor, right? Like the flavor of our food isn't simply taste, it's a combination of taste and smell together. So we process those two things really similar. And it makes sense because anything you're putting in your mouth, generally that you're going to have those chemosensory input on your tongue for taste, but then also, the epithelium of your nasal cavities right there. So that aroma, I guess I'll call it, is going to go up there too. Yep, we're super closely integrated. That's why if you lose your sense of smell, eating kind of sucks. I've never had that happen, fortunately, but you just can't, like everything kind of loses its flavor. One of the symptoms of COVID that I hope none of you have suffered from. Oops, and finally the insula, that's where we're processing taste, more specifically, as well as general visceral sensations. So remember those visceral sensory input is things like information from your digestive tract, could be something like nausea, input about like how full your stomach might be after eating, how full your bladder is, and that kind of stuff is processed in the insula. So all of these, what I want to emphasize here is that all of these lobes of the cerebrum are include the cerebral cortex, and that's that surface of the gray matter that I talked about earlier. Remember, that's these neuronal cell bodies, dendrites, and short axons, generally unmyelinated. It's only a couple millimeters thick, but because of all of these this convoluted surface area, thanks to all these sulci and gyri, It's actually like almost half of your brain's mass is this cerebral cortex. And I mentioned earlier just a little bit about the medical history. In the past, how they would know what areas are responsible for what function is that, someone would get bopped on the head in a certain area and then they'd lose the ability to do a certain thing. And they'd say, oh, that area of the brain must be responsible for this particular thing. Now, thanks to PET scans or fMRIs, functional magnetic resonance imaging, I think is what it stands for. You can actually see kind of where blood flow goes when you're doing certain things. And there's, do I have this in here? No. There's a diagram in the book that shows kind of the brain lit up under different activities. So when you're thinking, concentrating on something, in this case, you can see these areas involved with speech and hearing when a person is involved with those kinds of activities. So we can use these new technologies to better understand what parts of the cerebral cortex are responsible for what kind of activities. Yeah, go ahead. This is let's see. Yeah, so this is your. Yep. So this is looking like this way. So this is the frontal lobe, parietal, temporal lobe here, and then I can't quite tell the cerebellum would be here, but I think it's not even on here. I think this is just the cerebellum. Yeah, it's a little weird. I'm glad you asked that. So in our cerebral cortex, we have three kind of general functional areas. And those are the sensory area. So for each of our senses, we have what we call a primary sensory cortex. So an area that is mostly involved with processing that information. Then we have association areas, which basically integrate information from a whole different places and help us associate it, as you might guess. And then finally, we have motor areas where we can both plan and then actually initiate voluntary motor control. And what's kind of cool is those are separate areas. So there's an area, let's say that I want to do something really exciting, like pick up this water bottle, right? Part of my brain is going to say, okay, I want to pick up that water bottle. How am I going to do that? And needs to think about, I need to locomote there. So I need to take a step. I'm also going to need to extend my hand, extend my fingers, etc. And then that part of the brain sends that signal to another area that like initiates all of that and allows me to make this kind of coordinated motion with the water. At the same time, there's information processed in our cerebellum, bless you, that allows me to maintain balance. Basically, walking in humans and running is basically just controlled falling. Every time you take a step, you're basically like falling through space and catching yourself, which is why when you trip, like catch your toe on something, you fly face first, because each time you do that, you're kind of losing your balance intentionally. So the cerebellum is involved with kind of coordinating all of that and making sure that those muscle movements are happening in a coordinated way, and also that I'm maintaining my equilibrium as I'm doing all those things. So all of these areas of the brain have to work in conjunction with each other just to do something really simple, like grab my water bottle. Yeah, actually, I'm going to talk about, I think I got ahead of myself a little bit. Let me let you get this slide, see what questions you have. And then I think I'll do one more slide here because I already talked about it ahead of myself. Just get so excited about it. The association area is more about integrating sensory input. So what the association area would be is, let's say there was an orange here. That would be the area where the visual information, I was gonna say auditory, but oranges are fairly quiet. The olfactory information, maybe if I'm touching it, the tactile information all kind of come together. And then that part of the brain says, looks like an orange, smells like an orange, feels like an orange, must be an orange, right? And so the basic plan as we're engaging these parts of our brain, the basic kind of information flow is this, that sensory information is received by the primary sensory cortex. Again, there can be the visual cortex in the case of vision, olfactory in the case of smell, etc. And then that information goes to the sensory association area where it's all kind of integrated. And then there's certain association areas called multimodal that are getting information from all of these different senses. And then we can enact a plan, voluntary motion to respond to that. And all of this is done by interneurons in the cerebral cortex, in these sensory and motor areas. So all of these, it's all interneurons within the central nervous system relaying information back and forth. And I want to bring that idea back to this diagram that we talked about at the very, very beginning of the nervous system, the general flow of information. But now we know a little bit more. We know specifically as information is integrated, we know that this is happening in these association areas of the cerebral hemisphere. So you've basically seen this diagram a couple times now, but I want to just bring it back because now we know more detail about how that information is integrated. It goes to that primary sensory cortex, and then from there to the association cortices where it's all kind of brought together. Yes, good. Yep. And that'll be the last slide. That's where we're going to end off today. We'll pick this up on Friday. But let me let you get that information. Any other questions before we wrap it up? All right, so I think what I'll do, I'll just go ahead and skip to this last slide. I don't know why I put a cartoon on today, but I did. Just as a reminder, Danny will have a tutoring session tomorrow in the library. I will provide information. We'll have another quiz before Thanksgiving, and I'll provide information about that for you on Friday. I'll give you the details. And then I just have a ticket out with standard format here. We've been a little bit of a routine with the tickets out. I need to mix it up a bit, but I'm just going to have you upload your drawings and answer a couple questions about today's class. As you're doing that, I'm going to pass back your quizzes. I have these, well, you have your grades, but I want to give your actual quiz back. And let me know if you have questions about anything. Otherwise, I'll see you all in class on Friday.